Hello everyone, this is a review session on the models for the OSPE upcoming exam. So here we have the uterus, which is number 1 on the checklist, and here we have the fallopian tube. In detail, here we have the fimbriae, and then here we have the infundibulum, and then the ampulla, and then here is the isthmus, and then here is the ovary. And this in general from here is the uterine cavity, so where the morula enters from the fallopian tube into the uterus. And here we have the fundus of the uterus as well. And we also have the ovary ligament, which is basically a connection between the ovary and the uterus. And then we have the cervix of the uterus, which is this part in here. So right over here is the supravaginal, because it's the upper part of the vagina, right? Then if we continue, that's the cervix. And here is the lower part of the vagina. And here we have the endometrium. This lining is the endometrium, and then the myometrium, and then this is the perimetrium. The cumulus ovoris is basically the cluster of cells around the developing follicle, that we can say in a way that they are the early stage of corona radiata. Now, as the oocyte will continue to develop and mature and all of that, later on the cumulus ovoris will expand, forming later on the corona radiata right on top of it. So we can see the cumulus ovoris is in a way deep to the corona radiata. Now I just want to show you guys that this is the mature graphene follicle with the secondary oocyte, and we're gonna study what is basically going on here. As you guys can see, we all know that what is placed here is the antrum, right? And then from here we have the granulosa cells, and here in blue, whether it's light blue or whatever, this is the theca interna, and then the skin-like from here, the outer part, is the theca externa. Okay, so this represents the fertilization stage. This represents the 2 cell stage. This represents the 4, and this one represents the 8. And then from 16 to 32, this represents the morula. This is the stage where the fertilized zygote, in a sense, enters the uterus. And then here we have the blastula, and this is gastrula, because there is like the bilaminar disc is forming in here. And also you guys need to remember that in this one is the embryoblast, and whatever is in here is the trophoblast. Okay, so since this one is the embryoblast, this is the blastocele, or another name for it is what? The blastocyst cavity. And then since here it differentiated already, the epiblast, and here is the hypoblast. How do we know this is the epiblast? Because it's high columnar, and the hypoblast is cuboidal, right? So the epiblast is going to later give us what? Ectoderm, and hypoblast is going to give us endoderm. And now, since this is the amniotic cavity, in the roof and on the walls of this cavity we have amnioblast cells, or another name for them is the amniogenic cells. And in here, basically, this is where the embryoblast is starting to implant. And in general, not the embryoblast, the whole thing is being implanted into the endometrium. Okay, so in here, we have the amnioblast cells, and here the cytotrophoblast. And then whatever is embedded into the uterus or into the endometrium is the syncytiotrophoblast. It's not really shown in this figure, but I just wanted to give the general idea. Also, the little strings in here coming out, this is the chorionic villi. Now for the extra embryonic, splanchnic and somatic mesoderm. Now whatever lines the amniotic cavity from above, this is called the somatic extra embryonic mesoderm, and as well the trophoblast. And whatever lines the yolk sac from the outside, that is going to be the splanchnic extra embryonic mesoderm. This is the connecting stalk. This just shows the transition from a primary yolk sac to a secondary yolk sac. And how can we know? Is because the sac itself gets smaller, and we can see it being constricted to the connecting stalk. Now, the trophoblastic lacunae, by the trophoblast, there will be invasion of maternal blood vessels to get into these type of lacunae, so that they can be filled in with blood for the embryo. Now, the maternal vessels are not really shown in any of the pictures nor the models, so I just tried to show you guys that the trophoblastic lacunae are going to basically be the early maternal vessels just invaginating there to feed the baby with the blood. Now, in the pictures, the uterine gland is shown, As you guys see in here, it's basically a gland that is in the endometrium itself. And its responsibility is basically to secrete uterine milk and the nutrients to the developing embryo. To be able to see the exocoelomic membrane and the exocoelomic cavity, we will have to view it from this picture. So it's basically the primitive yolk sac is the same as the exocoelomic cavity, and the membrane is the little cells that are lining the yolk sac. It has another name as well. And here we also have the fibrin coagulum, which basically seals the implanted embryo at the implantation site. Here we have the primary villi. As you guys can see, it's these little small cells that after that will later on develop into secondary and then tertiary. And here we have the buccopharyngeal membrane, or we can see the oropharyngeal membrane. It's the same. You see the primitive streak, and here is the primitive node. So this is the placenta. As you guys can see, from here we have the maternal side, and whatever is above is the fetal side. Now, here are the maternal blood vessels. This is the uterine wall. This one is the decidua basalis, and then this one is the chorionic plate. And from the chorionic plate, we have extending the chorionic villi. And here we have the fetal capillaries, so arterioles and venules and all of that. And the background is the intervillous space. All right. And here we have the umbilical arteries and veins, and then again, this is the chorionic plate. And here we have the umbilical arteries and veins extend into the umbilical cord. So right here we have the embryo. This is the primitive eye, and here we have the primitive ear. These are the primitive pharyngeal arches. Here we have the somites development. Here we have the upper limb bud and lower limb bud. And then here we have the coaxial bud and the umbilical cord. And here we have the cardiogenic area, and here's the liver prominence, which is caudal to that part. So what type of questions can they ask us? They can simply ask for the labeling of the structure. So what is the name of it? They can ask of the fate. So for example, the yolk sac, it will be later on changed to a primitive and a definitive yolk sac. They can even ask for the embryological periods in weeks. So when did this structure develop? Clinical procedure performed in a specific area. So if they're pointing out to, let's say, the amniotic cavity, and they can ask us what type of procedure that can be done in here. It can be, for example, amniocentesis. Congenital abnormality related to the labeled structure. So if they did point out, for example, on, let's say, the amniotic fluid, for example, yani, and they can ask us what type of congenital abnormality is related to it. So what we can have in our mind is what oligohydramnios and polyhydramnios. Maybe they will give us more hints related to that to help us identify. Now, state the function of a structure. This is most commonly asked. And the significance. So if there's a specific structure that has been made, they want to know what is the importance of it. So what is the solution? To review all of our embryology lectures. Now I understand that we have to study the manual, the pictures that are included as well, but the embryology lectures will help to cover for us the majority of what can be asked, and it will give us an idea of how the exam will be. Here we have some examples. Okay, so what is the label of number 1? We know this. This is the amnion. Fate of structure number 6. So this is the uterine cavity, and we know as the fetus continues to grow, the cavity will keep on getting smaller and smaller until it is completely filled, and it will disappear as the chorionic cleave, amnion, and decidua capsularis, they will fuse together. And state one of the most common congenital abnormalities related to structure number 4. Structure number 4 is the yolk sac. We have learned many, but the most common one is what Michael's diverticulum. And for this picture, here we have the placenta. Label the structure number 1. This is the umbilical vein, and these two in red are the umbilical arteries. Which structure contributes to the utero-placental circulation? Now, utero is related from the mother side. Placental is the fetal side. So in here, they're basically hinting to the space that helps to exchange like we can see the nutrients, the waste between those two sides. And it is basically the intervillous space, which is structure number 2. The function of number 4. These are the maternal blood vessels. They help to deliver oxygen and nutrients to the fetus and remove any waste that has come from the fetal side. Here are more examples that you guys can practice on that I have seen from certain documents that have been collected by students. Credits to them. And you guys can practice on these. Just use the same questions that I have wrote here. Two things that I probably forgot to mention is that they can do get us, unfortunately, questions related to genetics and physiology. By physiology, I mean the hormones that are secreted during oogenesis. And in relation to the genetics, probably the mitosis and meiosis process, all of that. So do try to get over these topics as much as you can. And inshallah, everything is going to be fine. And good luck. Thank you, everyone.